So let's talk about the eukaryotic cell cycle. So we had already mentioned that the cell cycle can be broken down into a number of phases. The M phase, the mitotic phase, and interphase, where the cell is growing and copying its chromosomes in preparation for dividing. So mitosis and cytokinesis then are going to be the most active components of the cell cycle with regard to what we can actually see under a light microscope. So again, if we look at a cell coming right out of the M phase, entering G1, and G stands for gap. So this is the first gap. This is when if you were observing a cell, it wouldn't look like much was going on because everything's happening on a molecular level. The cell is growing, it's making resources, it's kind of getting ready to make a copy of its DNA. And then it will enter S phase. And in S phase, it will actively then copy its DNA by all the mechanisms we had talked about before. And so in essence then it has doubled the amount of DNA in the cell. It has made a copy of its genome. And then it would move into gap 2, G2. The G2 phase now is the last opportunity for the cell to create all the materials that it's going to need to get through cell division, the actual physical distribution of the nuclear contents and dividing the cell in two. Because once we enter mitosis, M phase, we really have condensed our chromosomes now into packages that are easier to move around, so we've kind of shut down gene expression. We've got to have all the proteins ready to roll that are going to do stuff to help us get through this. So again, if we look at that, interphase is going to be about 90% of the cell cycle. G1, S for synthesis, and G2. So the cell is growing and active in its own right during this portion, but again, under a microscope, a light microscope, you're not going to see a whole lot going on. But then, of course, we're going to move into mitosis, the M phase. And mitosis can be broken down into 5 phases, and I really combine prophase and prometaphase into a single phase in my mind. But the easiest way that I remember this is I say PMAT. So P for prophase and prometaphase, metaphase, anaphase, telophase, PMAT. And that way it can help you remember these terms. Now, cytokinesis, the actual cytokinesis, the movement of the cell to divide, is going to start ambiguously somewhere in the last telophase, last portions of telophase. But what we want to do is look at each of these in turn. And again, you can see these under a microscope, a light microscope. So as we end G2 of interphase and move into prophase and prometaphase, what we're going to see is the nuclear envelope beginning to disassemble. The formation of what's called the mitotic spindle. So these are going to be microtubules that are going to be built to ultimately then connect to the chromosomes so they can be physically manipulated, so they can be moved around the cell and lined up in such a way that will then ultimately divide the two replicated chromatids from each other and kind of advancing them to full-fledged chromosomes. So we'll get to that in a second. So prophase and prometaphase, we see dissolution of the nuclear envelope, condensation of the chromatin into condensed chromosomes, and then attachment of those chromosomes to the mitotic spindle, which is also forming. Again, this is made of microtubules. Now, when we go into metaphase, we'll get them all lined up on the metaphase plate. Anaphase will pull them apart, and telophase will kind of reverse prophase and prometaphase. So let's go over these again because you're going to need to study these and need to hear them again and again. The main points from prophase and prometaphase are the chromatin condenses into our condensed chromosome format. The nuclear envelope is disassembled, and mitotic spindle is assembled and attached to the chromosomes. All right. In metaphase, we're going to line them all up on the metaphase plate. So remember, the mitotic spindle is composed of microtubules, right? And these are going to begin at centrosomes that are kind of located on opposite sides of the nucleus. That's important because ultimately we want to pull the split chromosome, the two chromatids, apart from each other in opposite directions so that we can split the cell down the middle. So we talk about the asters. These are short arrays of microtubules that extend from each centrosome, and we're going to attach that microtubule to the kinetic core of the chromosomes, all these C words. And that's going to be a protein complex that allows these two things to interact and actually move the chromosome down the microtubule. So if we look at this, we can talk about what's called the metaphase plate. And that's going to be the imaginary slice through the cell where all the chromosomes will align. equidistance between the two centrosomes. All right. So again, the cartoons are much clearer than the electron micrographs. So you can see in the cartoon how this kind of glass plate is cut through the cell. It's like you took a watermelon, sliced it right down the middle. That slice is where you'd have the metaphase plate. So we're going to line up the chromosomes on the metaphase plate, and then as the microtubules from the mitotic spindle connect, we're going to actually pull apart these two sister chromatids and separate them. So they're now individual chromosomes moving apart from each other towards the opposite ends of the cells. The microtubules are actually going to be depolymerized at the kinetic core end. So we're actually munching away at that microtubule as we move down it towards the centrosome. Now, at the same time, there are components of the mitotic spindle that are actually pushing against each other and pushing the cell into an elongated format, trying to separate what will be the two new nuclei. So that's anaphase as the chromosomes separate from each other. And then as we enter telophase, we kind of reverse the steps of prophase and prometaphase. We're going to see a decondensation of the chromosomes into their chromatin format and a reformation of the nuclear envelope, as well as then a breakdown of the mitotic spindle. So if you go over this thing enough, and get it in your head, it's not that hard. But again, some of the terms and phrases may be unfamiliar to you. Now, cytokinesis itself is going to be slightly different between animal cells and plant cells. So in animal cells, what we have is what's called a cleavage furrow that forms. And this is literally there are cytoskeletal elements that form like a noose underneath the plasma membrane and then cinch it together like a sausage, separating the two daughter cells. Now, in a plant cell, you have the cell wall to consider. So what you do is basically build a new cell wall between the new two daughter cells. And that's called the formation of what's called the cell plate. All right. So you should be able to look at pictures of the different phases of mitosis, the M phase of the cell cycle, and identify them. So again, know what prophase and prometaphase are. Again, I'll probably lump those two together. Metaphase lined up along the metaphase plate, anaphase separating, and then telophase, kind of the reversal of prophase and prometaphase. Again, remember that cell plate formation is going to be the big difference between animal cells and plant cells in this process.